So, hi everyone. I'd like to welcome everybody to the webinar. My name is Holly Bonar, and I'm a technical application specialist with DeNova Software. So today we're going to be talking about the stain index. So what is it exactly? How is it calculated? And how FCS Express can help you use the stain index to optimize your experimental design. And so just a couple of housekeeping rules for the webinar if you haven't already and you can hear me and see my screen. Say hello in the question box there. And as we go along, please feel free to put questions in that question box in that GoToMeeting control panel here. My colleagues Greg Veltri and Sean Burke are also on the webinar with me, and they'll answer those questions as we go along. Okay. So let's go through an outline of our talk today. So first, we're going to talk about what the stain index is, and this will include some discussion about how it's calculated and how it can help you with your experimental design. I'll show you some of the tools that are available in FCS Express that can be used to calculate the stain index, including using our integrated spreadsheets that allow you to do all of your calculations directly in the software. We'll also talk about using the stain index to do antibody titrations, and I'll show you our universal antibody titration templates. They're already built and available for download on our website, so you can quickly get started titrating your own antibodies. And then finally, at the end, I'll show you just a few more examples of some other methods for calculating the stain index in FCS Express and what you might use that information for. So as the number of fluorochromes and available fluorochromes increase and become brighter, experimental design has become more and more important to the success of a flow cytometry experiment. And so now we need some strategies for figuring out the best antibody fluorochrome combinations so that each population can be separated out appropriately. And so this overall brightness of a fluorochrome is determined by the relationship between the positive and negative populations of your staining, as well as the background signals of stained cells. And so in these background signals are affected by several factors. And so it can include things like the signal intensity, autofluorescence, nonspecific staining, and the electronic noise of the instrument. So how can we normalize all of these factors to come up with some idea of how bright our fluorochrome is? And so this is where the stain index comes in. The stain index is a normalized signal to noise ratio. And so this calculation takes into account a few different things. It takes into account the separation between your positive and negative staining populations. In other words, the distance between where these two peaks are. And then it also takes into account the spread of that negative population, or the width. So to calculate the stain ratio, you can take this distance and divide it by the width. And what that means is that you're taking the median of the positive peak, subtracting the median of the negative peak, and dividing it by 2 times the standard deviation of the negative peak. And so what does this mean from a practical point of view? It means that when you see a high separation between positive and negative populations with a low spread of the negative population, you'll get a high stain index number. And then if you see a decreased separation between positive and negative populations, or an increased high spread of that negative population, you will get a low stain index number. So if we take a look at the 3 histograms that we have here, these are all the same. These are cells that have been stained with the same antibody, only that antibody's been conjugated to different fluorochromes. And so in the first histogram, you can see that our negative population is nice and narrow, but the positive population isn't super bright. And on this middle histogram, you can see that our positive population is brighter than the first, but we also see increased spread here in our negative peak. And then finally here on the right-hand side, we've kind of got a combination of the 2 things we're looking for. We've got this nice bright peak here, and we also have got limited spread in our negative population. So if you were to go back and do the stain index calculation, you would see that these numbers jive with what we're seeing here. This histogram has the lowest stain index, and this one has the highest. And so now we have a way to kind of quantify that relative brightness of our antibody fluorochrome combinations that's taking into account the things that are going on with the instrument we're using and of those background signals of the environment we're staining in. Okay. So now this doesn't mean that an antibody combination with a high stain index is good or one with a low stain index is bad. So it's just an indication of the relative brightness of that antibody combination. And so why do we care about this information? How do we use it in our panel design? So if we're determining the relative brightness of a series of fluorochromes, we can use the stain index method to kind of help simplify our experimental design process. So if we have the high density antigens such as CD3, CD4, CD45, we can use antibody combinations that have a lower stain index. And that's because there's enough antigen on the surface of the cell to be able to separate out those populations, even if the signal is dimmer. And conversely, if we have a low density antigen such as CD19, CD25, or cytokines, we would want to use an antibody with a higher stain index. And that way we can pull those populations out with a brighter fluorochrome when we don't have a lot of antigen to bind to in the first place. So let's take a look at how we can easily calculate the stain index in FCS Express using our integrated spreadsheet tool. And so the spreadsheet tool allows you to do your flow analysis and calculations without having to export those numbers out to another software like Excel. So let me pull up this layout here. Here we go. So in this layout, we're calculating the stain index for several different concentrations of antibody. And so this is going to be kind of the first step or the first part of an antibody titration experiment. And we'll talk about why antibody titrations are important more in a little bit. So for now, we're just going to see how we can calculate the stain index. So here we have a number of different positive controls where the cells have been stained with various and increasing concentrations of CD3 APC Cy7, right? And so we've already gated that out. So you can see our dot plot up here where we're looking at forward scatter versus side scatter. And we've gated our lymph population. And then down here, we've drawn color dot plots where we're looking at the positive and negative populations of CD3 APC Cy7. And so our positive populations are green and our negative population is blue. And we've drawn a gate around both of those. Okay. Up here at the top, we have a histogram overlay of all of our data files. And so you can see that with our positive population, as you increase the amount of antibody, you do get a brighter signal, which is what you're looking for. But you can also notice that as you increase that amount of antibody, you're also seeing increased spread here in the negative population. And so remember, it's not just about what is brightest visually. We're looking for the right antibody and the right amount of antibody that gives us the best separation between these two populations while minimizing this negative spread. And so we can quantify that by calculating the stain index. Okay. And so we're going to do that using our integrated spreadsheet. And so we've inserted one down here already, and you can insert them pretty easily by going to our toolbar up here, choosing the Insert tab, and then clicking the New Spreadsheet button. And so in this spreadsheet, we've already entered our concentrations of our positive controls, ranging from 15 nanograms to 1,000 nanograms. And then in the rest of the spreadsheet, we're going to put in the statistics that we need in order to calculate the stain index. And so I've listed it down here. Here's our equation. So we're going to need the median of the positive peaks, the median of the negative peaks, and the standard deviation of the negative peak to calculate the stain index. And so the great thing about our integrated spreadsheets is that you can go to the data list and pull all of these statistics from all of the data files at the same time. So we'll go up here to the data list, pull that out, and we can see all of our tubes here, and they're all there and in the correct order. So the first statistic that we need is the median of the positive peak. So I'm just going to type here, median in the positive, and then I can take all of my data files in the data list, highlight them, and drag them directly into my spreadsheet and drop them. And so the first window that pops up is asking you what type of token are you putting into your spreadsheet. So we're going to choose statistic token, click OK. And then it's going to ask us for the details of that particular statistic that we want to put in there. So we want the APC Cy7 parameter, because that's what we're looking at. We want the positive population, And then we also want the median. And we'll click OK. And you'll see that all of the positive peak medians have been dropped for all of those data files automatically into our spreadsheet. And so then now we can repeat that for our negative median and our negative standard deviation. And so the neat thing about this is that if I'm generating statistics from the same gate, I can choose them both at the same time. So I'm going to take my files again, drop them directly into my spreadsheet, choose statistic token. Again, we want the APC Psi7 parameter. This time we want the negative gate, and then we want both the median, and then I'm going to hold down the shift key and choose standard deviation. So this is going to pull both the median and the standard deviation statistics for our median or negative population. And then I'm going to click OK. And now you can see that we have the statistics for both of those in the spreadsheet. And you could pull as many statistics as you want, and it will drop them sequentially into your spreadsheet. Okay. It also generated the column names for us since they were both coming from the same gate, and that saves us just a little bit of time. Okay. So now we have all the statistics for, close that, for everything that we need to calculate the stain index. And so we're going to do that in our fifth column here. I'm just going to type in stain index. And we're going to start with this first cell here. So we can enter the formula directly in this first cell the same way that we would do it in Excel. And so if you've ever typed formulas in Excel, you'll know that everything has to start with an equal sign. And you can get to the formula a couple different ways. So if you go up to Spreadsheet Formulas in your toolbar, you'll see that there's a number of different types of formulas that you can insert into a spreadsheet. And you can see here that it's nearly every formula that you could find in Excel, right? Even some of these don't even have anything to do with flow cytometry. So there's financial, there's logical. The stain index is found in the statistical grouping. And so they're listed in alphabetical order. So you can always come down here to this button and scroll if you don't know the name of it. Or if you know the name of it, or you don't want to take the time to scroll through it, you can simply come here, start typing the formula that you want, the type index. And then when the software realizes what formula you're looking for, it will automatically kind of pull up this block of information here that tells you how this formula is calculated, okay? So we have our stain index formula, and I'm just going to follow the list of statistics that it's calling for and put them in exactly the same way I would do an Excel spreadsheet. So the first statistic is the median positive of the population. So I'm just going to come over here and click that cell where that value is, and then hit a comma, and then it'll go to the next one. So now I need the median of the negative population. We'll insert that one. And then finally, the standard deviation of the negative population. There we go. Closing the formula with parentheses, just like we would in Excel. And then hitting enter, and then you'll see that the stain index has been calculated. Now we can extend this calculation to the rest of the values that we have in our spreadsheet, same way you would in Excel, by clicking on the cell and dragging it down. And so now we have all of our stain indices for all of our file types. Okay. So now that we've calculated all the stain indices, we can add a chart to visualize the data. And for this particular data is titration data, so we're going to insert a regression plot to look at it. I'm going to highlight my spreadsheet and go up to the Insert tab. And this is where, again, the Insert tab is where you can find all of the different plots, from anywhere from dot plots or plots to visualize your flow data to Excel type plots like bar charts and regression plots and things like that. So I'm going to go in the other section. You'll see a plot that's called Scatter with Regression. I'm going to click that, and we'll draw it right here. And with our integrated spreadsheets, all of the columns now become options for our X and Y axes of our plot. So for example, if I click on the Y-axis, you'll see that I have concentrations, the two medians, our standard deviation and stain index, and these are the columns that are in our spreadsheet. So I can choose any one of these to show on our axis. Since we're doing titration data, we'll look at stain index on the Y-axis, but we'll leave concentration here on the X-axis. Now the black dots are our data points. And so you can see that we get a nice exponential increase of our staining that plateaus out as you get to our higher concentrations. right? And it's this plateau that you're looking for when you're doing antibody titration. This is a regression plot. So our regression analysis has already been applied here. So that's what this red line is. And we can come in and make some changes to this plot. So you can see this data is kind of smashed up here at the top. So we can change this axis to kind of make our data look a little bit better. So over here on the right side is our formatting window. So if I want to change this axis and maybe bring it down just a little bit, I'm going to go to axes, and I'm on my y-axis. And instead of this being automatic, I'm going to change this range to be from 0 and let's say 25. And so now you can see we've brought that plateau down just a little bit more so you can see it. I also want to change our red line regression here. So the default for our regression plots is a linear regression. But since we're doing titration data, we don't want a linear regression. We want to do a Michaelis-Menten regression. So you can see in our model options here, we have this drop-down menu, and we have a lot of different algorithms that can be applied to this regression analysis. We're going to choose Michaelis-Menten. You can see now our red line kind of fits our data quite nicely. And it generates the Vmax and all of the other statistics that come with this regression. So it generates the Vmax and the R-squared value. And then the second part of the titration would be to go on and do the calculation to figure out what your optimal antibody amount is. And we're going to do that here next. Okay. So why are antibody titrations important? And so we've got this regression curve. And so what are we going to do with this? So antibody titrations allow you to determine the optimal concentration of antibody to use within your panels in order to obtain that best resolution of your data. And so it's a really crucial component of panel optimization. It's going to help reduce antibody aggregation and background noise and buffers. It's going to minimize opportunities for non-specific binding. It enhances resolution of those low, mediate, and moderate and highly expressed populations. And it's also going to decrease costs and increase your experimental efficiency because you're only going to be using the amount of antibody that you need rather than maybe what the manufacturer might recommend. So FCS Express offers many tools that allow you to set up a standardized and reproducible titration analysis for calculating this volume. And you can do it all within the same software package. And what it's going to allow you to do is create a regression plot looking at stain index versus increasing amounts of antibody. And that's what we did in that previous analysis. And then when the data plateaus, or when you reach 90% of Vmax, somewhere in this area is your optimal amount of antibody to use, right? Because you want to use the right amount of antibody that's going to give you the best signal, but not use too much of it. Because at some point, you're never going to get a better signal. So our antibody titration templates, we've created in FCS Express in order to facilitate and simplify the analysis of your titration data for our customers, right? So if you don't want to go back and build this all on your own, because you could always create it manually, we've already done it for you. And so everything is already built into the template. Once the data is brought in, the user just adjusts the parameters and modifies the gates around their specific data set. And then all of the calculations needed for determining the appropriate amount of antibody to use are automatically updated in real time. So the templates are going to calculate the stain index, and this includes both the standard formula that we've been talking about in this webinar, but it's also going to include something called the 84th percentile method, which you may have heard of before. And it's also going to calculate the standard signal to noise ratio that's not normalized. And so, and we did this to give you options in terms of choosing the best method that fits to your data. It's going to generate all those regression plots from our integrated spreadsheets, and it's going to calculate that 90% of Vmax. And then it's going to take all of those calculations and extrapolate what the optimal antibody volumes are to use using that data set. So once you're done analyzing your data, batch processing can be used to export the data, either for documentation purposes or for presenting to another lab or to another group. And since we know that not all antibody titrations are designed exactly the same way, we do have 5-point, 6-point, and 7-point titration templates that you can download directly from our website to start using immediately. Okay. Okay, so let's take a look at this 7-point antibody titration layout. Let me open it up here. And so I already have some data loaded into this template. And you can see over here in our data list that all of our data files have loaded correctly. And we've loaded them, the template is designed so that you load them in from the lowest titration to the highest titration. And this is important because the order is going to determine how the statistics are calculated. Okay. And so when you first look at the data that's loaded, you can kind of see these 3 columns of plots that are already here. And so the first row is our scatter plot that looks at forward scatter versus side scatter. In our second column, we have our positive and negative staining populations. So on our x-axis, we're putting the antibody that we're titrating here. In this particular data set, we're doing CD3V7. And then this third column over here is the negative population that's going to be used to calculate that 84th percentile control for that other method that I talked about. Okay. So the first thing you need to do is you're probably going to have to adjust your scatter gate and your positive and negative gates appropriately, right? Because everybody's data set looks different. And so a good method for doing this, since we've got a lot of plots in here, is that you can right click on the top plot, come down to maximize, and it's going to make this plot bigger so it fills your screen. So you can really kind of see what's going on. And you can see that we've just got a generic gate drawn here. We've got a rectangle gate around our population. So you can adjust this in a couple of ways. You can either adjust your gate around it accordingly, or you can just redraw it if you need something that's going to fit your data better. So to do that, you could go to the gating tab, choose polygon, and you can just start drawing a new gate right on top of the existing one. Okay, I'm going to come here, connect the dots, and when I do that, the create new gate window pops up. But instead of creating a new gate, we're going to choose replace an existing gate. And the gate we want to replace for this one is the scatter gate. And so this is a really nice feature in the software that allows you to recreate gates that maybe need to be adjusted or changed. But so you're not having to go back and delete everything and start over just because you want to change what your gate looks like. So we'll click OK. And you'll see that now my rectangular gate is gone, and we've replaced it with this nice custom gate that we've drawn here that fits our data better. Okay. So when you're done with that, you can right click, choose restore, and it will go back to where it was. So all the gates in FCS Express are universal gates, meaning that this one gate is going to look the same on each of these plots, even though there are different data files for each one of them. And a lot of times that works out just fine. And it keeps you from having to draw individual gates for every single data file for every single plot. But there are times when you do see some scatter differences, or you do see some positive staining differences between data files, and you want to adjust your gate just for that data file. So you can do that by changing this gate to a data-specific gate. And so to do that, you would come to the plot that you would want to change. So for example, this one looks like it needs to be adjusted a little bit. So I'm going to highlight this gate, and if I hold down the Shift key, I can now make changes to this gate, and it will turn it into a data-specific gate. So you can see now that this gate is specific to this data file, and all of the rest of the gates have stayed the same. And I can come to each one of these plots, hold down the Shift key, and adjust this gate as I need to. Okay. So now we're going to do the same with our negative and positive staining. So I'm going to come up here, right-click, go to maximize. You can see, again, I've got these two kind of generic rectangular gates here. And I might want to come in and draw something that's a little bit more custom to what my data looks like. I'm just going to draw a polygon gate around my negative population. Again, I'm going to replace it. This one is for negative staining. I'm going to click OK. And then my rectangular box disappears and my new gate replaces it. And then I'm going to do the same for my positive population. Okay. Replace it, choose positive staining, and click OK. And so now you can see I've got everything gated nicely, and it conforms to the shape of this population. and I right click, choose restore. And so again, these are universal gates, so everything looks the same. But if I come in here and I want to adjust this one dot plot, I can hold down the shift key, change it, and it stays specific for this plot only. Okay. So now that we have our gating strategy complete, so there's a few more items that we need to address on this template. So over here on the left-hand side, at the top, we have some demographic information that has already filled in. These are all tokens, all information that's pulled from the metadata of the files that we're analyzing. So the acquisition date of the files already pulled in by itself, the acquisition instrument, the current date, and the person who's doing the analyst, or the person who's analyzing the data, all shows here automatically. For this token down here, we need to insert the antibody that we're titrating. So for this data set, it is CD3B7. And this information is going to be used when we do our batch actions in a bit to generate our reports. And then in this token, we need to enter the concentrations of each of the files that we have in our data set. And so for this data set, the names of the files actually has the concentrations in here. So I'm just going to use them from here. And again, we're going to go from lowest amount of antibody used to highest amount of antibody used. So I'm just going to start typing here. And the important part of this is that you only want to put a number in this box. If you're titrating with antibody data where you've used a certain amount of antibody, so for example, in here we're using microliters as our units. Other data sets uses the amount of antibody, so like nanograms of antibody. You don't need to put, or should not be putting the units into these boxes. You just want to put the number, and that's because the number is going to be used in the calculation. If you put the units in there, the formulas won't work. Okay. So before we move on, we're going to take a quick look at this whole page to make sure everything looks correct. So we see all of our plots have data. They're all in the right order in our data list. All of our gates look good. Everything's gated appropriately. And then over here, we've got all of our concentrations in and the name of our antibody that we're titrating. Okay. So once we've done that, we can go to our stain index page. And you can see that all of the plots and all of the calculations have updated automatically. And so these are all happening in the background as we gated our data and as we put in those concentrations. And so when we were done, everything is going to snap into place. Up here at the top, we've got our two stain index calculations. This first one is just a regular stain index calculation that we've been talking about so far. The second one is using that 84th percentile method. And you can see here that we've got our R-squared values, we've got our red line regression curves, and they look pretty good. Down here on the bottom left, we have a straight signal to noise ratio calculation. And so an unnormalized signal to noise ratio is simply the median of the positive peak divided by the median of the negative peak. We have a nice R-squared value here as well. And then for this plot, we're just doing a correlation between these two stain index methods to see how similar they are in the calculation. And so here you can see we've got this nice straight line and a real nice R-squared value here, kind of showing that these two methods aren't that different. In this middle spreadsheet here, you can see our calculations. So we've calculated the 90% of Vmax, and then have back calculated and extrapolated out what the best concentration amount of antibody it is to use for this particular data set. Okay. We also have a list of our fit statistics here with our R-squared values that look really good. Our bottom spreadsheet contains all of the statistics that were used in doing these calculations in these plots here. So this spreadsheet looks very similar to the one that we showed in our first example, where we had all of our positive and negative median peaks. We had the standard deviation of our negative peak, our concentrations, and then anything else we needed to calculate those other plots in here as well. Okay. So once you've done your analysis, we've also set up batch actions so that you can easily save this layout and create a PDF and a PowerPoint for your documentation files. So if you aren't familiar with batch processing, this is the method that FCS Express uses to automatically perform a series of actions once your original analysis is complete. And so we do have other webinars and some other resources on our website that will show you how to build those from scratch if that's something you're interested in doing. So I'll show you what we already have built here. So we go to the batch tab and choose batch actions. And in here, we've put in here that we want to export this layout to PDF. This way we can save it for documentation purposes. We're going to save it, and it's going to save the layout linked with all the data, location of all the data. So if you go to open this layout 6 months from now because you want to look at it, it will automatically pull the data files and populate the template automatically, so you won't have to do that again. And then we've also set it up to export to PowerPoint. So if you're doing a series of titrations and you want to take them to a lab meeting and present to your group or show people what your titrations look like, you can easily do that. So once you have your batch actions added here, we can close this, and then we're going to click the Run button. And it's going to pull all the data and create those files automatically. Okay. There we go. And so here's our PDF. So you can see that our plots are all here. All of the demographic information is here. If you scroll down, you see everything nicely. It also has our plots, our spreadsheets with our calculations and our antibody amount. Okay. You can see here at the top, we have our PDF or our PowerPoint presentation. And when you export layouts to PowerPoint in FCS Express, it will save all of the information that's in your layout. So you can see that we have our plots here and our notes that were in the gray space, and then you can see everything that's on the white space as well. And this looks exactly the same as what it looks like in your layout. And you can go from slide to slide, look at the different plots and the calculations and the spreadsheets, calculating spreadsheets down here. Okay. If you were to present this in presentation mode, you would only see the items that were on the white space of the PowerPoint, but it does still retain anything that you have in gray space. Okay. So I do want to talk about a few other features of this layout. So what I just showed you, I had already populated some data in it to show you what it looks like. But when you first open the layout, the very first thing you'll see is the startup script. And it's going to look like a dialog box, just like this. And we put in a startup script into this layout as a way to streamline bringing your data in, because this is a universal antibody titration template. So it's meant to be used by a lot of different people who are in a lot of different platforms that may have a lot of different data sets. And so the startup script is how we've made it easy for people to bring in data from all those different types of platforms, right? And it's going to walk you through some of the questions so that the software can find your data, load it, and set the correct parameters for the analysis before you even get to the gating part of it. And it's just going to make it that much easier and save you some time. So the first screen of the script tells you to read the titration template information page before proceeding. And so that's the page that's directly behind the script, so you'll be able to see that. And you can just kind of move this screen a little bit out of the way so you can read all of the instructions here before you get started. So I'm not going to go through all of this individually, but it's a good idea to read the information before proceeding if you haven't used the template before. So it has some specific information about how to use the script or how to turn it off if you don't want to use it. And then it has some information about some instrument-specific settings that might need to be changed, particularly if you're a Cytech user. So you can just move this out of the way, check out this page, make sure everything's set the way it needs to be. And then you can kind of go through the questions of the startup screen to get your data populated, right? The other thing I want to mention is the last page. The whole titration template is actually 4 pages. It has that first informational page that will actually be hidden once you've loaded your data. And then it has a third page. And it's this third page that goes through some of the instructions for how the template was built and some instructions for the users. It contains a link for full PDF instructions for the use of the template. So you can click on this link, it's a live link, and it will pull the PDF instructions. So you can have that PDF with the instructions next to you on another screen. So you can just go through step by step for how to work the template. There's a link for some more information about the stain index on our website. And then it's got our email address here. So the template was designed to work with a number of different data sets, but if you have any problems with your data set or any questions about how the template works, you can certainly email us here and we'd be happy to help you with that. So don't struggle with anything, please just reach out to us. And then finally, we've got this table here that has a list of the formulas and calculations that we've used in the software. So it's got that regular stain index calculation. So we have media of the positive minus media of the negative divided by 2 times the standard deviation of the negative, plus this 84th percentile method. And then we have the spreadsheet formulas. So the stain index formula, we also use this percentile plot formula for the 84th percentile method. And then we also have the regression plot y to x, which does the extrapolation of data to calculate that optimal antibody amount to use. All right, so coming up on the end here, I just want to show you a few more examples of what you can do with stain index calculations in FCS Express. If you have any questions, again, just feel free to put them in that question box and we'll get to those as well. So this template is a basic global template that could potentially be reused anytime you want to calculate the stain indices for a series of antibodies. So on this layout, we've got 3 plots here. So we have our scatter plot, forward scatter and side scatter, looking at our lymph population. We have a color plot. We are looking at the same data set that we looked in that earlier template. So we've got CD3, APC size 7 here. So we've got our positive and our negative populations. And then we also have a histogram showing that same data. Here we've inserted an explanation of what our stain index calculation is. Again, it's that same positive minus negative median divided by 2 times the standard deviation. And then we've inserted this custom table over here. You can insert a table up here by our insert tab, text box, and then inserting a table. And then we've calculated the stain index in this layout using custom tokens. And so a custom token is a nice way to create any calculation you want that isn't already available in FCS Express in the standard list of statistics for your plots. So if we go to View, Custom Tokens, you'll see a list of the custom tokens we've created here. And so again, we've created all the statistics that we need to make that stain index calculation. So we have our positive median, our negative median, the distance between those two, so subtracting the negative median from the positive median. We have the negative standard deviation. And then for our stain index, we're taking the distance between the positive and the negative and dividing it by 2 times the standard deviation. And kind of the cool thing about the custom token is once it's built and it's in here, you can drag this anywhere into your layout that you want to display it. So just drag it right out, drop it somewhere in my layout. It can be on the white space or the gray space. And then it just becomes a text editable object in the layout. So you can come up here to text, change the font if you want something fancy, make it bigger so people can see it, change the color, space it out, resize it, however you want. And so this can be a really nice feature when you want to show your statistics in places that doesn't necessarily look like a spreadsheet. Okay. We'll close this. If you wanted to do this calculation in a spreadsheet, you could. I've entered it here, so now you can see the custom table method and the integrated spreadsheet method. Either one of them will work. Just kind of depends on what you want your layout to look like. Okay. And so for this layout, we're showing one data file at a time. So if we go to our data list, you can see that we actually have 7 different data files here. If you remember, each of these had increasing amounts of antibody. And we can cycle through our data just by clicking on the blue iteration numbers in our data list, right? So this first one is our lowest amount, 15 nanograms And as we go through, we're increasing, but we've added this overlay so you can see what the lowest concentration looks like. And then as you recycle through your data, you can see how that positive peak gets bigger, brighter and brighter, but also that negative peak gets whiter and whiter. Okay. We've also added some batch actions here. So for this one, we've set up an Excel report. So it's going to take all of these statistics that are in our custom token and export them out to Excel so that you could use them for whatever you want to use them for. We've also created a PowerPoint, and we've even created a batch action that's going to allow us to save pictures of our histograms individually. Okay. So again, we're going to go ahead and close this and run it. It's pulling all of the data, all of the items across all of the different data points, all of the different data files in our data list. And it will open automatically. And so here's our Excel spreadsheet. And you can see that this table looks very similar to that integrated spreadsheet table that we created in our first example. We have our positive and our negative medians, the difference between the two of them, the standard deviation, and then the stain index calculation. And so having this out to Excel, you could use it to store your data. You can append data to it. So if you ran another titer experiment, you could continue to add things onto the spreadsheet. You could use it to generate plots in here if you want to. Okay. And then finally, on our last example, let's say you have a bunch of antibody fluorochrome combinations across many different lasers, and you want to get a handle on what is the range of the stain index for each of for those combinations, right? So you have a bunch of antibodies, and you just kind of want to know where you stand. And so this is a layout where we have calculated the stain index for a number of antibody fluorochrome populations, or combinations, sorry. This is data from a 4-laser BD-LSR2 instrument, and we have all the data for each of these lasers. We've created histograms for each one of the fluorochromes, and we've placed markers over the positive and negative peaks. And this is so we can generate the median and the standard deviation statistics that we need to calculate that stain index. Okay. We've inserted a table here. So this is similar to our custom table that we created in the previous layout. We have our statistics here, and then we've used custom tokens to generate the stain index. Okay, we'll look at our custom token window real quick, and we've got them all for each of our lasers. Okay, and again, this is a 4-laser instrument. So if you look at the individual tabs, we have one for each laser, and you can see that we've done all of the calculations here. So on this final page, we've taken all the stain index calculations for all of our fluorochromes and kind of summarized them into these tables. And so you can do that with a custom table like we've done here. So everything's been reordered from the ones with the highest stain index down to the lowest ones. But we've also used our spreadsheet to generate this bar chart. And so now you can get this visualization of all the antibodies that you have and kind of where they sit in terms of the stain index, right? And so it's real easy to see that you've got some high stain index combos up here, some in the middle, and then some at the end. And so now you've got some, you know, in the past you may think, oh, you know, bright fluorochromes include things like PE or APC, dimmer ones include fluorochromes like FCS, but now you have some quantitative numbers and some quantitative assessments of that particular antibody with that particular fluorochrome on your specific instrument. And so it really just gives you a better handle on what those antibody combos are going to do for you. Okay. So just to sum up, we've been talking about the stain index as a method for determining the relative brightness of a fluorochrome. And it's going to take into account the difference between those positive and negative populations, as well as the spread of that negative population. And it's really useful in helping to optimize panel design is by helping users kind of figure out the best antibody antigen combinations, as well as helping determine the optimal amount of antibody to use via the titration experiments. And so I think we've shown FCS Express has many tools that can kind of help make those analyses really easy to create and use. And so things like integrated spreadsheets, custom tokens, formulas, The ability to insert regression plots directly in the software, so you don't have to go out to Excel if you don't want to, is really going to save you a lot of time and energy in the lab. And the really nice thing is that these templates can be saved and reused. So once you have an antibody titration template, or if you've downloaded our universal titration templates, you can simply save them as blank templates and then open them up and reuse them anytime you want. So you're not having to go back and recreate these analyses every time you have a new data set. And finally, thanks everybody for attending. Here at the end, I just wanted to let you know about a few additional resources that we do have. We do have free one-on-one training for getting started with the software. This can just be one-on-one help, or it can be with a small group. We can work with your own data to kind of help get you moving in the right direction with whatever it is you're working on. And so you can set that up by emailing us at support@denovosoftware.com, and we'll be happy to get you moving that way. We also have, if you don't already have the software, we also have a free 30-day demo that you can try out. It's a fully functional demonstration version that has all of the features turned on and available for you to use. You can download that from our website, denovosoftware.com. And we're also happy to do those training sessions for you with that demo. So you don't have to purchase the software to get help from us on using the software. Also on our website, we have a page with more information and some application examples. So this is that link that I showed you earlier where you can find the universal titration templates. If you don't want to write down this link, you can also click the magnifying glass and search for flow application samples. So you'll get any sort of templates that we've uploaded for you to use, but we also have videos and other written tutorials to kind of help show you how to do some of these things. And so again, anytime you have any questions, either help with the titration templates or any other questions you might have, email us at the support@denovosoftware.com anytime, and we'd be happy to help you. So thank you again for your time today. If you have any other questions, we'll keep the window open for a little bit to answer any last minute things. And thanks for attending.